Hello everyone, welcome to the Knowledge Hub by Sri Chaitanya. This is Sayyid Danish Anjum, and today in this session we are going to start with the next phylum, that is phylum Coelenterata or Cnidaria. So in the previous phylum we have studied the basic characteristics along with some examples of phylum Porifera. So in this session we will continue with the next phylum, that is phylum Coelenterata or phylum Cnidaria. Now as we can see that it includes diverse animals like hydra, jellyfish. Let me take the pen first. So it includes diverse animals like hydra, jellyfishes, sea anemones and corals. Corals are of interesting value and corals are of interesting example. We will see that how does the corals makes the coral reef. And apart from that, previously this phylum was termed as Coelenterata, and it included the cnidarians along with the ctenophores. But now, as of now, the Cnidaria are classified as separate phylum and ctenophores are classified as separate phylum. Okay? Now, at present, the ctenophores have been assigned as a separate phylum status. Although, if you look the anatomy of the phylum Coelenterata and phylum Cnidaria, both of them are having similar anatomical features. There are differences, there are some characteristic properties, so due to which the ctenophores are now assigned as separate phylum. So we will see. Now, as we see the habit and habitat characteristic of this phylum characters or this phylum members, we will see that Coelenterata or Cnidaria are exclusively aquatic in nature. But when we talk of aquatic, again as in the phylum Porifera we discussed, aquatic means either in marine or either in freshwater. So here also aquatic means that they are having at both the habitat, that means marine habitat as well as freshwater habitat. So when I say marine habitat, most of the Coelenterata or Cnidaria are characterized or are having habitat of marine aquatic form. And when we talk about the example of freshwater, then the most important example is the hydra itself, which is a freshwater species. So this is the habitat characteristic. That means they all are aquatic, but some are marine exclusively and some are in freshwater species. Now, when we talk about the habit, that means we have to understand about how do they spend their lives, whether they do locomotion or whether they are sessile, whether they like to live in the solitude or whether they are living in colonial form or in associated form. So here are two examples. The first one, the example right in front of you is the hydra, which is having a polyp form and it is usually sessile in nature. So that means most of its life it is spent as a sessile organism. What does the sessile means? That in the Porifera I have already described that sessile means that an organism through its body part remains attached to the substratum and it does not locomote and it does not show locomotion, right? And also it usually spends its life as a single solitude organism. So along with the sessile it is also showing solitude habitat. So I am writing here with the sessile solitude habitat, right? So it does not live in group, it does not depend on the other type to spend its life or to have other characteristics, to have other nutritional requirements or to find prey or to escape from a predator. It does not usually associate with the other types. But in contradiction, here we see that this is the medusa form. Most likely, most common example is the jellyfish Aurelia. So it is usually free swimming and it lives in colonial form. Now what does that mean? That in the colonial form it will be living in the group of its similar kind, and this is an advantage to the comparison of the solitude, because in group it's easier to find nutrition as well as it is easier to escape from the predator also. So these two are the habit and habitat points of the phylum Cnidaria, as we move further, they are aquatic, mostly marine, sessile or free swimming, and the most important character is they are having radial symmetry. So why they are having radial symmetry? Because if you see, they are having a cylindrical shaped body, and the cylindrical shape appearance of the body is usually having radial symmetry. So these are mostly marine, and a few like Hydra live in fresh water. Here we can see that this is the example of Obelia. It is also similar to the Hydra, and Hydra is a very unique freshwater polyp form. Now the polyp and medusa, we will also discuss about these morphological forms, but first let's have a look at the habits. Many are colonial. Example is the Obelia. Here you can see I have taken a picture from the microscopic view is shown of the Obelia, that is also called as Cipher, and here is the diagrammatic representation of the Obelia. So you can see that this form is not free swimming, it is sessile, it is remaining attached to some substratum or some solid place. Now in contradiction, if you see this one, this is the example of Physalia or the Portuguese man of war, and you can see the tentacles and its another gas filled chamber called pneumatophore, this one as you can see on the top of the surface. So it usually helps this organism, this gas chamber pneumatophore helps the Physalia to float in the sea, so it's a free swimming kind. So here we can see that this is how the Hydra looks microscopically, and this is another example of the polyp form sea anemone. So here we can see both. Now as we move, sessile Obelia or free swimming Aurelia. So this is the example of Obelia, Cipher, and this is the example of Aurelia, which is commonly also called as jellyfish, right? Now level of organization, in the previous phylum it was cellular level of aggregation, cellular level of organization, but in this case now the level has been advanced up to the tissue level of organization. So Cnidaria exhibit tissue level of organization and are diploblastic. That means they are having two germ layers, ectoderm and endoderm. Mesoderm germ layer is absent in them. Instead, similar to Porifera, which are having mesenchyme, they are having mesoglia, but that mesoglia is not similar in composition wise to Porifera. It is having different composition, and also it is having different types of cells in it. So overall three layers are there: ectodermal layer, middle mesoglia layer, and the inner towards the cavity is the gastral layer or the endodermal layer. So here we can see a diagrammatic representation. This layer in the orange part is the epidermal layer, this layer in the green part is the endodermal layer, and the middle one blue is the mesoglia. This is the middle layer. Now, as we can move towards the symmetry part, you can easily get an idea from this diagrammatic representation that how a cylindrical body organization is having a radial symmetry. In the previous sessions, I have also tried to explain you with the help of a cylindrical bottle that how does that radial symmetry is having in cylindrical organism, because at every plane we cut it longitudinally, we get identical half. So here we can see that multiple plane we are cutting the organism longitudinally, although we are getting each time identical half. That's why they are having radial symmetry. Sea anemones show biradial symmetry, and the body is radially symmetrical in most of the polyp form. Now talking about the exoskeleton, some of the Cnidaria, example corals. Now this is the important part. Corals have a skeleton composed of calcium carbonate, and due to this, when the organism usually settle down at the depth at the sea floor, it gets deposited as calcium carbonate, and due to this continuation process of deposition, there is a rock formation of calcium carbonate termed as coral. So the term which we usually see as coral reef is nothing but it is the deposition of the corals, which are organisms having deposition of calcium carbonate. Okay, so certain anthozoans. What are anthozoans? As we will proceed towards the end of the chapter, we will see the classification of Cnidaria. So in there are three classes: anthozoa, scyphozoa, hydrozoa. Out of that, one class is the anthozoans, and a few hydrozoans in which epidermis of polyps secrete a calcareous or horny exoskeleton and are called as coral. So in this diagram we can see that the polyp form is secreting calcium carbonate and due to this, when it gets settled down at the floor, it usually deposits as a calcium carbonate rock. So their exoskeleton is also commonly called as coral. Now, as we move further, we can see the coral reef. Here we can see the example, although the picture is not very much clear, but it provides a habitat to many other numerous multi-species. So it's of important significance, ecologically important. Coral reefs are of very important, ecologically significant rock and ecology. They create an ecosystem for many other diverse type of species. So coral reefs are the large formation of calcium carbonate laid down by certain corals in shallow tropical seas. Now, you must remember that when we talk about the corals, it's not just the cnidarians who are having coral. Many other phylums are also having. Many other phylums are also having corals examples in them. So ultimately, when those organisms gets deposited at the surface in the form of calcium carbonate, they get deposited. So they ultimately, all those organisms, all those corals from other species also, they also contribute toward the formation of the coral reef. So you must keep that in mind. So Great Barrier Reef is the largest coral reef in the world. Now, as we can see, the body form and the body wall, it is composed of mostly two cellular layer, that is the outermost is the epidermis and the innermost towards the cavity is the endodermis layer or the gastral layer. So as we have seen in the case of the porifera, that there also we have studied two layers, the epidermal layer, pinacoderm, and the endodermal or gastral layer, which we called it as guanoderm. Pinacoderm made up of pinacocytes and guanoderm made up of guanocytes. So similarly, we can see here that there are two layers. The outermost layer is composed of different cells and the innermost layer is composed of different type of cells. So usually the outermost type of cells we called as epitheliomuscular cells. So why we call them as epitheliomuscular cells? Epithelium is towards the epithelium, that is the epithelial layer, and muscular because this epithelial cell has basically two portions, the apical portion which is towards the outer surface and the basal portion which is towards the inside or towards the cavity surface. It is having a contractile protein called myonin, so which is similar to a contractile muscle protein, due to which it can shows the phenomena of contraction. So that is why it is called as epitheliomuscular layer. And in between we also have sensory cells. Apart from it, we also have other type of cells called as interstitial cells. So these other type of cells have different functions. We will see what type of cells is having what type of function. So starting with the body wall, it is composed of outer epithelium, the epidermis, inner epithelium, the gastrodermis, this one. So gastrodermis layer is also composed of many type of cells, out of which the most common are the endotheliomuscular cells, similar to epitheliomuscular cells, and also some gland cells which will secrete something mucus or something digestive enzymes. And apart from that, sensory cells or interstitial cells will also be present in this gastrodermis layer also. Now, if we talk about the middle gelatinous layer, we will see that this layer is the middle gelatinous jelly-like layer, which is also present in the phylum Ctenophora, although it is transparent there. But here this layer contains other type of cells, and when we talk about the other type of cells, we are emphasized on the particular type of cells called as neurons, or which are creating nerve net like structure. So here we can see the outer epithelium, the epidermis, the inner epithelium, this is the gastrodermis. Let me labeled it correctly. Gastrodermis, this is the epidermis, you can also see in the diagram similarly, and the middle layer is the mesoglia, gelatinous. Mesoglia often contains amoeboid cells derived from ectoderm germ layer. Because ectoderm is also having interstitial cells. Remember archaeocyte cells in the porifera? They were used to be totipotent cells, totipotent stem cells. So this is interstitial cells which are also present in the ectodermal layer. They are also the similar type of totipotent cells, and they can give rise to any other type of cells, and hence mesoglia often contains amoeboid cells derived from ectoderm layer. Epitheliomuscular cells and nutritive muscular cells are contractile in nature. I have already explained why they are contractile, because of this contractile protein present at the basal part of the cell called as myonin. Now here we can see the diagrammatic representation that this is the mesoglia, this is the endoderm, that means it is present towards the cavity. I will show you the cavity also. And these are the gland cells, these are the ciliated cells, so endodermal epithelial cells. Ring muscles is present also at the basal part. Longitudinal muscles is also present at the basal part in epitheliomuscular cells. And here we can see nematocyte. This is the special kind of characteristic cell. We will study about it in the upcoming slides. So ectodermal epithelial cell, interstitial stem cells which are totipotent in nature, nematoblast cells which are again nematocytes, nidoblast type of cells which are used in offense and defense, and sensory neurons which will sense the external stimuli and transfer it to the nerve net like structure present in the mesoglia. So this is a common diagrammatic representation. Here you can see also the ganglionic neuron, but in this diagram nerve net is not shown. We will discuss it in the upcoming slides. So moving further, as we can see, the body wall and the cavity which is called as gastrovascular cavity. Why it is being called as gastrovascular cavity? Because gastric cavity, digestion will take place extracellularly here, and also vascular means the circulation of the digested food particle will also be there. So together it is termed as gastrovascular cavity. It will be present both in polyp form as well as in medusa form, as you can see the marking. Now in polyp form, this is the body stalk and this is the mouth part. It is present on a special structure in Hydra, particularly called as hypostome, and these are the tentacles. So here are also tentacles present. Gastrodermis layer you can see in the inside, and mesoglia in the middle, and outer one is the epidermis. So this is the oral end of the organism at which the oral part or the mouth part is present, and the opposite end is called as aboral end. So here oral part is here and aboral part is opposite. This is how we distinguish the oral aboral end. Mesoglia is thin in polyps and it is thick in medusa form, in which it is important in providing buoyancy force. It is well developed in the members of Scyphozoa and hence called jellyfishes. So Scyphozoa members are also called as jellyfishes. We will see the classification also in the upcoming slides. Now, mentioning about the important characteristic cell of this phylum is the nidoblast or nidocytes. Here we can see that nidocyte is the characteristic cell of this phylum. Similarly, let me write it down. So it is the characteristic. Similarly, as we have studied in the porifera, that the characteristic cell was choanocytes or collar cells. So here the characteristic cells are the nidocytes or nidoblast. So the name nidaria is derived from the nidoblast or nidocytes, which contain the stinging capsule or nematocyst, which is also filled with toxin, sometimes usually called as hypnotoxin, and it is present on the tentacles and the body. So it is usually present at the outer surface. In the inner side, in the gastral layer, nidocytes are not present. You must remember this point. So at the outermost layer and in the tentacles part, this nidocyte is present. Now this particular cell is commonly called as stinging cell, and it is a common offense and defense weapon. So it is used to capture the prey, it is used for locomotion, it is used for anchorage. So each nidocyte contains a fluid-filled membranous capsule called nematocyst. So as we can see in this particular diagram, that this example, particularly this one, right now, this is the example of undischarged nidoblast. Now this one, when it gets discharged, it usually erupt out all of its part along with its needle thread-like structure, and with this needle thread-like structure, it also releases the hypnotoxin which is present inside this capsule. So hypnotoxin is filled in this capsule, and once it is ejected out with its thread point, it also releases that hypnotoxin into the prey. So it usually the kind of knife part, that knife it's usually open out, it gets stuck inside the prey, and it's a needle-like structure, injection-like. So through that injection needle-like structure, the hypnotoxin is released into the prey body. So as we can see, this is the microscopic real-time image, and we can see that how does this thread-like structure neuroblast get discharged, and this needle thread-like structure gets erupted out, and with that pointed end we can see that hypnotoxin is also getting released. Now you can see that this is the discharge and this is the hypnotoxin being released from each needle, from each thread, right? So this is how the neuroblast gets discharged. So this is the figure, you can see this is the example of undischarged neuroblast, and the adjacent one is of the discharged neuroblast. So this is how the neuroblast gets released, its inner content. Now it usually gets released, gets excited when there is a trigger or a stimulus from the atmosphere, from the outer environment, or when the prey comes in contact with the tentacle, then the neuroblast of the tentacle, they suddenly releases their inner content via the thread into the prey, and they can kill the prey or hypnotize the prey. So the fluid usually is called as hypnotoxin, and this particular capsule, this one which you can see, this particular capsule, I am marking it with red, this particular capsule is called as nematocyst, which is a fluid filled containing capsule. So whatever the fluid will be there, it gets releases through this particular thread like this. So this is a fluid filled structure, and when the neuroblast get discharged, the fluid get released through its thread like this one. Okay, so moving to the locomotion part in Hydra. Hydra uses different type of locomotion, as you can see, like looping, gliding, walking, and also somersaulting, like this one. So these are the different locomotion as shown by the Hydra. Although it is in polyform, although it is sessile, but it also shows locomotion from some time or from time to time, depends upon the condition. So when we talk of an organism that is sessile, then by characterizing it as a sessile, we means that the organism, it's for majority of its life, it remain attached to a solid substratum, and it remain sessile for most of its time period. So that is the meaning of sessile characteristic. But some of the sessile remain confined to their position for lifelong, and some for majority part, and they will move or show locomotion when it is required, right? Now moving to the gastrovascular cavity or coelentron cavity. In Porifera it was paragastric cavity. In here it is gastrovascular cavity or coelentron cavity. What are the characteristics of coelentron cavity? Like in Porifera, the paragastric cavity or the spongiocele was just a cavity in which there was no digestion taking place, no extracellular digestion was there, but in here the digestion will be extracellular as well as intracellular. First it will be extracellular and then it will be intracellular. So they have a central gastrovascular cavity with a single opening mouth or hypostome. On hypostome. So if you can want to understand what is mouth being on hypostome, you can imagine that this is the face, this is the hypostome, and this is the mouth opening which is present on this hypostome. So hypostome is a structure on which the mouth is present. Like if you want to imagine, so this is particular structure is called as hypostome. Elevated structure on which mouth opening will be present, and the digestive tract is of incomplete. That means it is having, all the members are having blind sac body plan, right? So one end is open, the opposite end is or aboral end is closed, and only single opening is there both for ingestion and egestion of undigested food. Ingestion for the food and egestion for the undigested food, both from the single opening, right? So the name coelenterata refers to the presence of coelentron cavity. Mouth serves for ingestion as well as for the egestion, and coelentron helps for digestion as well as circulation. So this I have already explained. Now moving to the further next part, here we can see that this is the mouth, these are the tentacles, and digestive enzymes are released from the gland cells which are present in the gastral layer, and so the digestion takes extracellularly first, afterwards the partially digested food is phagocytosis by the cells of the gastral layer, and the further digestion is carried out in the intracellular manner. So digestion is both extracellular as well as intracellular, and first extracellular in the coelentron and then intracellular in the nutritive muscular cells of gastrodermis, right? So moving to the next part, respiration and excretion. Exchange of respiratory gases and elimination of excretory waste, which is the chief nitrogen containing excretory waste, is ammonia, and it occurs by the diffusion through the body wall. That means through water it will be directly exchanged, both the gases as well as the excretory substance ammonia. Now, this is the very interesting characteristic which appears first in the phylum Cnidaria only. Before it, phylum Porifera was not having any kind of nervous system. In fact, Porifera was not having any kind of system, but here the beginning of the nervous system like structures or the nervous system has been shown or has been seen in the phylum Cnidaria members. Neurons are interconnected to form a pair of nerve net. This is the important term, nerve net. And also to be emphasized here, neurons are not the kind of multipolar neurons in which we used to see in our body, but they are of the most simplest and primitive type of neurons. Mostly apolar type of neurons are there, which are present in the mesoglia layer, and through interconnection, through network, they form a nerve net like structure which is present in the mesoglia layer, and it is present in whole body, right? So it is not concentrated to a particular part, and one in the epidermis and the other in gastrodermis. Nerve cells are non-polarized, that means they are having a apolar neuron type structure. Also, nerve impulse conduction is diffuse conduction. Impulse can travel in any direction. So if a stimulus comes to this particular neuron, it will spread to other type of neurons in non-directional dependent manner, or it will diffuse to other body parts also. So if a stimulus comes to this part, then it will spread to rest body part also. So this is the diffuse type of nervous system. Now, sensory structure like statocyst occurs in the medusoid form. For example, tentaculocyst in jellyfish. Now, what is a statocyst? Student, for example, in our body we are having ear, and in ear we are having balancing part also, balancing organs also, which usually tells us our position in respect to the 3D environment. So similarly, these organisms are also having a structure statocyst, which tells or which helps them in maintaining their position or their body's equilibrium with regard to the environment. So this is the statocyst, is the balancing structure which is used to balance the body with respect to the environment, outer environment, right? So next, before moving to the next slide, I would like you to scan this QR code and to get registered with us, so you can know more about the courses and you can know more about the curriculum part of our institute, right? So moving to further, polyp and medusa are the two body form found in this member of phylum. Now this characteristic is particularly called as polymorphism also. Poly means many, morphism means morphological appearances. So if the organism in its lifetime is showing two morphological forms, two body forms, then we say that the organism is exhibiting polymorphism. So what are the two major polymorphic structures here? Polyp and medusa. So here is the polyp form, the cylindrical form, usually sessile. Here we can see the example of hydra. It is the example of budding hydra, but we can see that the hydra is having a polyp structure like this. And the other example is the medusa form, which is a free swimming form, and it is an umbrella shaped structure. And here we can see that the movement of the jellyfishes is taking like this only. So this particular part, this particular structure, okay, let me mark it with another pen. So this particular structure is the body part. These are the tentacles. So with the help of tentacles, it swims, freely swims. So this is the medusa form. Now what are the major parts of the What are the major differences between the two forms? Let us highlight those features. Polyform are common examples are Hydra or Adamsia or sea anemone. They are mostly cylindrical, they are sessile, they are solitary or maybe colonial in some cases. Mouth and tentacles faces upward like in this manner. Hypostome bears mouth, I have already explained. And mesoglia is thin. Nutrition, defense and asexual reproduction are the common characteristic of this form, although sexual reproduction is also found in polyform. So we will discuss it further in case of Hydra also. Now, in Medusa, for example, Aurelia or commonly called as jellyfishes. Now, umbrella or bell-shaped structure it is there, free swimming, solitary or colonial also, mouth and tentacle face downwards. Manumbrium bears mouth, another structure on which mouth is present. Mesoglia is thick and sexual reproduction is the common form of reproduction in the Medusa form. So it is also of dispersal type. Now, as we move towards the next polyp and Medusa form, then many type of individuals called zooids occur in some colonies, example Physalia. So this phenomena is also called as polymorphism. So as we can see, this is the example of Physalia. I have shown you in the GIF image in the previous slide also. It was having a pneumatophore, that means gas filled chamber, which was helping this organism to float on the surface. These are the tentacles bearing nematocyst, and these are the gonozoids. So smaller dactylozoids, gastrozoids, these are the different forms of polyps only, having different type of functions, which is not included in our syllabus right now. And as we can see, this is the another example of the similar form, having different type of polyp forms. Now, as we move further, those cnidarians which exhibit in both forms exhibit alternation of generation, that means metagenesis. So we must not get confused with the term metamerism. It is metagenesis, that is polyp produces Medusa asexually and Medusa forms the polyps sexually. Common example is the Obelia, which is also provided in the NEET NCERT textbook. Both asexual and sexual forms are diploid in nature and only haploid cells, certainly gametes, are formed. So such alternation of asexual and sexual phase is called as metagenesis. So let us see that what is the main thing happening in this form. So this is the polyp form, right, this one, okay. These are the polyp form and this is the Medusa form. So what happens, the polyp give rise to Medusa asexually. Here we can see the budding form, Medusa bud, okay. So through the budding, small Medusa releases, and when the Medusa fertilization, Medusa do sexual reproduction, the gametes are released, fertilization takes place, zygote forms, and the further development through planula larval stage, it gives rise to the polyp form again. So this larva settle down and again it forms the polyp body. So it will be remain as attached to solid substratum. It will be called as sessile in nature, right? So the Medusa produce the polyp form sexually and the polyp produces the Medusa asexually. So this alternation of generation is termed as metagenesis, right? So sperm and egg fertilizes externally only, so external fertilization, and the development is indirect because it is having development via larval stage, also called as planula, free swimming larva stage, right? So it should not be confused with alternation of generation found in plants. Now, regeneration, cnidarians like the poriferans are also having great power of regeneration. So they have a remarkable ability to regenerate. Interstitial cells helps them to regenerate because interstitial cells are totipotent in nature. So in the body wall they are present and they are totipotent cells. So when the fragment is when the fragment is produced in any form, so those fragments, if they are having sufficient amount of totipotent cells, they can give rise to whole part of the body or whole body. In such a case, we can see the example of transverse incision. So if we cut down Hydra into 2 pieces or more than 2 pieces up to a desirable size portion, then each piece will form the complete Hydra. So this is the transverse incision and this is the example of longitudinal incision. Both are producing different body or both are producing young ones asexually. So both will be clones, right? So old part and this is the regenerated part. Here we can see that when we carry out the transverse incision, the part, if you carry out multiple also, the part which is closer to the mouth will form mouth and the part which is closer to the base stalk will form the rest of the base stalk part. So in this way, asexually also, by the power of regeneration or by fragmentation, they can reproduce. Now, asexual reproduction takes place by budding, but in this case, we have to know that budding takes place on the surface, so it will be categorized as exogenous budding, not like the poriferans which are having exogenous as well as endogenous gamule budding. So if we see the animation, we can see that a small minute bud first outgrows in the form of an outgrowth. Later on it gets developed, it develops tentacle, it develops mouth, and it gets separate from the rest of the body and it gets attached to solid substratum and it grows as a young offspring or young hydra. So it is similar or clone to the definitely its parent. So similarly, we can see that these examples are showing, these are the live figure examples. We can see that the young hydra is budding and is also having its mouth part develop tentacles. So sooner or later it will get detached from the parent body, it will get attached to the solid substratum and it will again continue to grow as an adult hydra, right? Now, so sexual reproduction, Cnidaria are generally unisexual, but some are bisexual. That means some species are monoecious, some species are dioecious, and fertilization is external or internal fertilization is also seen in some, and development is both indirect. Development is indirect and includes a free swimming ciliated larval stage called planula. We have seen just in the previous slide. So fertilization is external, in some internal, and it shows indirect development as the development is happening through a free swimming larval stage, free swimming ciliated larval stage called as planula, right? Now moving to the next part, some examples of NCERT written here: hydra, Polyform Physalia, Portuguese man of war, Aurelia, jellyfishes, Adamsia, sea anemone, Meandrina, important one, brain coral. Also we have red coral also in the form of corallium, and Penatula sea pen and Gorgonia sea fen. These two are also the colonial form examples. We must keep in mind. So here are the some representative figures also. Metridium sea anemone, usually it's of cylindrical nature, and Metridium as well as Adamsia, both are sea anemone only and having different species. So here we can see that Adamsia also show mutualistic or symbiotic relationship with hermit crab. So when it gets attached to the body of hermit crab, and when the hermit crab gets shows locomotion, it changes its place. So does the sea anemone changes its place. So this way they gets transferred from one place to another, and due to sea anemone, predator usually does not attack the hermit crab. So this is the way they both benefit each other, and in this way they show mutualistic or symbiotic relationship. Now corallium or red coral, I have already explained that the coral are having two examples, red coral and Meandrina, which is given in NCERT as brain coral. So here we can see the example and the body diagrammatic representation of the corallium. The other examples are Gorgonia, sea fen, which is definitely a colonial form. We can see that its structure resembles a fan. So in general common name it is called as sea fen, and its structure resembles a feather pen. So it's generally called as sea pen. So both are usually colonial form having multiple type of polyps, right? Now according to the classification, the Cnidarian members are classified into three classes: Hydrozoa, Scyphozoa and Anthozoa. So Hydrozoa, as the name indicates, the common forms are Hydra. Scyphozoa are having jellyfishes, and rest are characterized in Anthozoa. Now Hydrozoa are mostly marine and some are freshwater also, for example Hydra. But in Scyphozoa all are marine, and in Anthozoa they are all marine. Moving to the next characteristic, stages in life, polyp and medusa, they both show. So the example will be understood that the obelia will be also categorized in this stage, in this class, and medusa is the predominant stage, and Anthozoa only show polyp stage during their entire life cycle. And usually Medusa, for example the Crespidot with the true velum and a Crespidot without true velum, and the true velum is absent in the Anthozoa. So these are the classification characteristics. Apart from these characteristics, there are other characteristics and there are other examples also, but in our syllabus we can have only this much of information. Now moving to some examples as an exercise. So what we have studied in this session, some of the questions are present here. You can have a look and you can answer them. I will change the slide to the answers in the next slide only. Sorry. So here we can see the name Cnidaria is derived due to the presence of on the tentacles and the body. So you must know the different names: cnidocytes, cnidoblast. Cnidoblasts are used for anchorage, capturing of prey, locomotion. Cnidarians have a central gastrovascular cavity with a single opening called mouth on hypostome. Digestion in Cnidaria is both. But you have to mention that first it is which and second it is which. First it is extracellular, then it is intracellular. Some of the Cnidarians like dash have a skeleton composed of calcium carbonate like corals, meandrina, and two basic body forms of coelentrates are this. You can have a look and you can sense the answer. I should not dictate it. I should wait. Some more questions are there. Umbrella shaped and free swimming body form, what it is called. Dash is the example for the Medusa form. Alternation of generation is called what? An example for the Cnidarians that existed in both polyp and Medusa form. In metagenesis, the polyp produce Medusae and Medusa forms polyp. Scientific names of Portuguese manova. You must remember the scientific name as well as the common name. Both the names are used in questions in the NEET examination, right? So Gorgonia and Meandrina are common names of Adamsia, Gorgonia and Meandrina are like. So these are the answers. You can have a look. You can pause and you can have a look. So moving further, these two are some questions from the previous year examination. Although there are many questions from this phylum also, but we have taken for the reference we have taken two questions. Given below are types of cells present in some animals. Which of the following cells can differentiate to perform different function? So choanocytes, interstitial cells, gastrodermal cells and nematocytes. But now students, what happens here, due to the extra information, what happens, the student get confused in the options. So I will tell you that how the confusion arises. For example, given below are the type of cells present in some animals. Okay, so which of the following cell can differentiate to perform different functions? So we have seen in choanocyte case also, it gives rise to germ layers. So now the student can think, okay, the choanocyte can also differentiate to give rise to germ layer. Interstitial cell is the totipotent, so definitely it is going to differentiate into any other kind of cells of the body. Gastrodermal cell does not differentiate, it is already differentiated cells, and nematocytes are also the differentiated cells. So the correct answer here will be interstitial cells. Why? Because it is totipotent in nature and it is not restricted to some differentiation, a little differentiation, rather it shows complete differentiation and it completely can differentiate into any other cells of the body part. So the correct answer will be interstitial cell. Coming to the next question, which one of the following statement is incorrect? So mesoglia is present in between ectoderm and endoderm in obelia. And the second part is exhibit radial symmetry, asteriasis, which is the correct one. Asteriasis means starfish. So starfish, we will see that in Echinodermata it shows pentaradial symmetry, so it comes in the radial symmetry only. Fasciola is a pseudocoelomate animal. Now it is Fasciola hepatica is a platyhelminthes. It is a acoelomate organism, not a pseudocoelomate. And Taenia is a triploblastic animal because it is correct. Why? Because it is having ectoderm, mesoderm and endoderm. Both the three germ layers are present. Mesoglia is present in between ecto and endo, that is correct. Exhibit radial symmetry, correct. So the correct answer will be fasciola. It is a pseudocoelomate animal, although it is related to the next phylum, upcoming phylums, but still we can have a look that how does the question can have options regarding from different phylum. So mesoglia is present between ecto and endo, that comes from the diploblastic category, Cnidaria and Ctenophora. Exhibit radial symmetry again from Echinodermata, and fasciola is a pseudocoelomate animal that comes from the Platyhelminthes. Pseudocoelomate is from the next phylum, next to next phylum, which is Aschelminthes, and Taenia is a triploblastic animal again from the Platyhelminthes. So the correct answer will be the fasciola, which is a pseudocoelomate, which is actually it is a acoelomate, but here we have to show the incorrect. So this is the incorrect statement. Okay, so these were the examples that can come. So to get to know more of us, to get to register with us, to know more about the courses, you can scan the QR code, get registered with us. You can also reach out to our nearest center, and stay tuned for the next session. In the next session, we are going to discuss from the Ctenophora.